Welcome to the Data Science webinar series organized by Manav Human Atlas Initiative. Myself, Shailesh Kumar, moderator of today's talk. Here, I would like to inform all participants that the total session time is 1 hour, where the talk will be of around 40 to 45 minutes. And then we will have a question answer session of 15 minutes at the end of the talk, after the completion of the talk. I request all participants to use question answer tab at the bottom of your screen for putting your questions. I request participants to put their question in the question answer tab at the bottom of their screen only. You can put your question in between the talks, but we will take after the completion of the talk during the question answer session. Okay. Now, I would like to introduce to our speaker for today, Dr. Sudhakaran Prabakaran. Dr. Sudhakaran is currently working as assistant professor at the University of Cambridge in Department of Genetics. He and his team is working on the use of data science approaches to prioritize disease mutations and to investigate the functional significance of mutation in non-coding region of the genome. It is really very interesting area, and I know Sudhakaran since last 5-6 years, and he's really uh, very interesting in the area in which he's working is really very interesting. He's also a fellow of St. Edmunds College, University of Cambridge, and also worked as a faculty of IISER Pune earlier before joining Cambridge University. Now over to Dr. Prabakaran.
Thank you, Shailesh. Thank you very much for this introduction. And I just want to thank Gayatri and Dr. Nagaraj and the entire Manav team as well for giving me this opportunity to talk to you all. So I hope everybody's doing fine. With that, I'm just going to jump into my talk so that we can cover a lot of ground. But I just want to make sure my screen is visible. So I'm going to just quickly share my screen. Is my screen visible? Shailesh,
yes. Yeah, it is visible. You can start presentation. Yeah.
Okay, cool. Thank you. So as Shailesh said, we use data science approaches to understand consequences of mutations in both coding regions and in non-coding regions. So that's what we primarily do. I mean, I don't work in COVID, but I've been following the literature and doing my own small projects. And besides that, I just want to declare that we're not actively working in any pandemics or anything, but we know a lot about data science approaches being used for genomics. Okay, so with that, I'll just start off with this simple history. So it's always a question of what did we learn, right? So whether it's biology or physics or any field. So let's just revisit what happened in the throughout the history of humanity, at least the recorded history of humanity. So there were five major pandemics. I hope you all understand the difference between pandemic, endemic, and epidemic, but I'm not going to go into that. So the first so-called pandemic was called the Plague of Justinian. It was around 540 CE, around 50 million people died. That was followed by the Black Death or the bubonic plague that killed 200 million people in just 4 years. So that's pretty much almost half of the European population, and probably a lot of people from Asia and other parts of the world too. But if you notice, this is the first time that people actually learned something, and this city called Venetian Ragusa in Italy. It's a Venetian-controlled port city, so they realized that if you isolate sailors that are arriving from other parts of the world, you can probably stop the plague. So they initially had this for 30 days, isolation for 30 days, and they call it Trentino. And they increased it for 40 days, and then it became Quarantino, and that's the origin of the word quarantine. Now, plague never really left Europe. It kept coming back and back again and again every 20 years. So this is the third pandemic happened in London around 1665. So that's the time when people started completely isolating people who were infected, and they were putting these red crosses, and that's the origin of the Red Cross. And followed by the around 18th and 19th centuries, people were moving from Europe and then to the New World, and brought in smallpox, and the smallpox decimated the entire Native American population. Around 95% of the people died. So what in 1796, Edward Jenner made this observation that people who milk people who work with cows, cows when they get cowpox, they don't suffer from smallpox. And he made this fundamental observation, and he took cowpox active viral particle, active whatever pus particles from cowpox, and infected his gardener's son, who was 9 years old, and he showed the world that if the kid was reinfected, when the kid was reinfected with smallpox, he didn't get it. So that was the first vaccine that was developed. So this was Edward Jenner's around 1796. But for us, for our purposes, the thing that that started off all these data science approaches to solve public health problems, it starts with this last pandemic, that is the cholera that happened in 1854. So what actually happened? That's what we're going to talk about here in the next slide. So John Snow, Dr. John Snow, he was one of the founding fathers of epidemiology. He was, one of the people who actually came up with anesthesia and the effects of anesthesia. But besides all this, he was a little puzzled with this cholera epidemic. At that time, they didn't know what was causing cholera and how to solve this. This was in London. A lot of people were dying. So he just took all the data points. He just took all the newspaper cuttings and all the maps, and he started putting things together. And he observed around 500 cases of cholera in one street in a district called Soho in London. This is like prime district in London. And what he observed was, you're seeing a big major red dot. So all these deaths happen in that one street around one pump. One pump is the pump that they use to pump water. So he immediately realized probably there is something to do with water or contamination of water. And then he observed the neighboring streets and neighboring pubs that served water. So at that time, people were drinking beer, and they claimed that beer doesn't give you cholera, but he proved that the beers that the people were brewing were using different pumps and different source of water. And he was able to narrow down to only one pump. And he proposed a simple solution, and the solution was just to remove the handle of the pump, and then the epidemic just panned out and slowly died out. So this is the power of data science. So this is the first time when clustering of data points was used to show that you can solve problems, biological problems of public health. And that is the start of all these observations. So from data, you observe patterns, and from patterns, you infer something fundamental. And with that, I just want to come to this particular slide. This is something which is very fundamental, and that happened in the field of physics. So the same thing happened. If you notice, Tycho Brahe came in and observed all these planetary motions, and he collected a lot of this data. And it took around 100 years or so for Johannes Kepler to come in and find patterns in these data sets, right? And another 100 years or so for Newton to come in and infer laws from these patterns. So that's what we are trying to get, because we have data, we have patterns, we have to infer laws, and we have to offer solutions, right? So is biology there yet? That's the question, and that's what we are all trying to understand. Is biology too complicated or too simple, or do we have enough data to infer fundamental laws? So let's see what we learned. This is the data set from the 1918 Spanish pandemic flu. Is there something that we can learn from? Is that something that informs us how to deal with this pandemic, right? So if you notice the brown bars in all these are all different cities, data from different cities, the number of deaths per 100,000. And this indicates the number of people, the isolation period. The brown blocks indicate the isolation period, and in some cities the isolation period was they had this isolation and social distancing measures taken earlier in the pandemic. In some places they implemented it later, and you will notice immediately, for example, in cities like New York. Where is New York? Here, the isolation period was for a long time. It was implemented earlier, and you see the disease died off, and there was no spike in the, there was no secondary spike. Whereas cities that were too early to implement, for example, Philadelphia, and or too late to implement and implemented for a short period of time, they had multiple spikes and they had increased deaths. So this is informing us something about pandemics and how to deal with pandemics, right? So have we learned anything from there? So we'll see what's happening currently with the COVID-19 spread. So this is data from this organization called endcoronavirus.org, and it's real-time data. This is acquired just yesterday. And these are the trends that they recommend for different countries. And you will notice immediately, sorry, the resolution is really bad, but this is the trends for different countries. And this is the US, trend for US, United Kingdom, and India. I was not able to pull out India and put it here, but India is also in the red here. It's kind of slowly increasing. And what we are noticing here is it looks like there are different trends for different places, and we simply have not learned anything from the past patterns of pandemics, right? Because each country has dealt it in a different way, and it's facing the consequences. And even the developed countries have not been able to implement anything properly because their trends are very in the negative region. So what's happening in India? So this is again trends collected by a private organization, and you will notice that some states have good trends where the green ones, where the disease is actually the spread of disease is decreasing, whereas in some states it's increasing, right? So different states have implemented different models, and they have different densities of people. So we are trying to figure out what is the right approach. And all these things can happen only with looking at this kind of data sets and trying to find patterns, right? So what I'm trying to emphasize is from data, how do we infer patterns, and from patterns, how do we infer solutions or laws? So you can do that in multiple ways, right? I think you must have heard Pranay talking about how you can model using mathematical and differential equations to understand the spread of this disease and figure out R-naughts and how to stop those R-naughts. Now we can use statistical modelings as well, statistical based inferences, and we can use machine learning approaches too. So the purpose of this talk is to give kind of a brief introduction on what machine learning is and how it is applied in the genomics field and what is happening in the COVID field and where people are using it. Okay? So what is machine learning? Machine learning is just simply looking at patterns, and looking at patterns, you infer some trends. For example, here, this is kind of the talk response I usually get, and I, any talk I give, not necessarily this particular talk. So I know this, and I know exactly what the equation is because it's not like I sat down and calculated every time. It's because I've observed this pattern many, many times, and I'm able to infer this curve. And if I actually infer details from this curve and even fit a proper gamma distribution to it. And have I sat down and calculated this? No. What have I done? So that's what I'm going to talk about. How did I come up with this curve? So you must have, this brings us to the big data world and how it is relevant to biological sciences and how it is being used. So there's an explosion of data sets in every field. It's not just in biological sciences, in economics, in history, in geology, and climate sciences. So everywhere there's this huge explosion, right? So how are we dealing with this kind of data set? And especially in biological sciences, you must have seen these graphs many, many, many times. The cost of sequencing has really come down, and the cost of computer storage and processing power and speed of processing power increase, cost has come down. So all these things are happening, and this has enabled us to implement machine learning approaches. And these machine learning approaches are very computationally intensive, and that is why with availability of data and with availability of computational resources, we are able to come up and implement new machine learning models. So this is something that I wanted to you all to take home. The message is we are living in great times where we can actually use all these data sets and do our own computations and infer patterns, right? So you all might be knowing, if you're all from the biological sciences background, every aspect of this cell, DNA, chromosome, transcriptome, proteome, we can scan them, we can get the expression profiles, and we have these different expression profiles even for the same cell. And we've been acquiring this day in and day out, and we've been collecting all this information, right? Copy number variation, gene expression levels, DNA methylation, microRNA. So what are we doing with this? So, and where are we heading? We are actually heading here. Pretty much, we crossed around 3 exabytes of data just for biological sciences, and that compares like it is kind of almost all the data sets in YouTube and Twitter and other huge, for example, people who collect the observatories, cosmological observatories that collect data. So some biological sciences have pretty much crossed all these observations, the amount of data that we've collected. So what happens with all these things? Data overload, right? So how are we dealing with this? So that's why we are in this sweet spot in history where we can implement many of these machine learning methodologies that we've been developing over the last 100 years or so on this new data sets that's becoming available and trying to find patterns. So example of one data set that's available. This is the TCGA. You must have heard of the PCOG as well. These are huge cancer repositories. This is an initiative of the NCI, National Cancer Institute in the US, where they have collected data sets from 11,000 patients in this growing repository. And this is made available to any researcher. And this is all childhood cancer data sets. That's 55 petabytes of data sets, and TCGA is more than 3 petabytes of data. And these are all these data sets are available. And another major data set is the Psych code, which we use all these data sets in the lab, and that's why I'm talking about them. This is all schizophrenia, bipolar related data sets that are again publicly deposited and made available to researchers. So how do we access all this petabyte scale data, right? So the traditional method is, even if you have a 10 Gbps internet speed, and if you want to download 2.5 petabytes, it'll take 23 days, and you need $1 million or $2 million to store them and computers to process them. And that was the old model. And each researcher is accessing these data sets individually, writing their 50 MB code, taking that code to the data, and then doing some calculations with that thing. But this has changed now. So you see what's the problem. Data gets locked away, cost is more, collaboration is prohibitive, and it requires a lot of computational resources. Now, all these repositories that I talked about, I also said they make their data available. How are they doing it? So they're making this data available through a cloud interface, right? So all this data is sitting in one central repository in the cloud. And researchers take their programs and run it in the cloud and find some patterns that they're looking for. So this is what has enabled us to embrace machine learning and biological sciences, because of these developments in the cloud computing world, and as well as an explosion of data sets. And you guys should all be excited about this, because in the previous scenario, you have to take your, it was very difficult to access these data sets because they were all siloed away. Now anybody and everybody can access these data sets and find patterns, right? You can individually sit in any part of the world and do that. So I'm going to just briefly talk about what is machine learning and give you different aspects of machine learning, just tell you what it is, and probably there's some take-home message for some of you who are interested in this. So what is machine learning? As I introduced to you a little earlier, by just looking at the data and just, I was able to plot that gamma distribution, right? So machine learning is just that. It is arriving at an hypothesis by just looking at patterns in data sets, and you're trying to predict something, right? So statistics is basically trying to find correlations between data points. Machine learning is trying to find some patterns and predict based on those patterns that you infer. Okay, so that's the difference at a very superficial level. But machine learning means you're not writing explicit sets of instructions for the computers to find patterns. You're just saying, okay, this is the data set. Can you tell us something? Okay, so there are multiple ways to do that, and one is supervised. There are different categories of machine learning approaches: supervised machine learning approach, unsupervised, semi-supervised reinforcement learning. For the purpose of this talk, I'm just going to stick to supervised and unsupervised. So supervised is when you have two labels, right? A, X, and Y. X is like gene expression, and Y is I don't know, some condition, and Y2 is another condition. So if you label and say this is one condition, this is another condition, and X is gene expression, that is supervised learning. Unsupervised learning is when you don't give any labels and ask the computers, ask the algorithms to find patterns. Okay, so this is the fundamental difference. And there are two approaches of machine learning, and this is the fundamental difference between those two approaches. So as I told you, machine learning has been there. It's been there for a long time, in the last 100 years or so, and it borrows from a lot of fields, from mathematics, computers, science, and statistics. For example, we have matrix algebra, we have calculus, we have optimization, and we have kernels, we have hashing. So that's why it's called a field. That's why it's called a science, and people call it data science too. It's a lot of things that come together, and there's a lot of optimization going on. There's no one software, one approach, one methodology. So having said all these things, I have to tone my rhetoric down a little bit. What this figure is showing is although we are living in great times and we can implement a lot of complicated models, machine learning is still nascent, meaning we're still struggling to figure out whether the image that we are seeing here is a muffin or a dog, right? It's still not there yet, even with all the developments that has happened. And you will notice that in 1958 itself, people were talking about computers will be able to walk, talk, and write and reproduce. And this is 1958, and we are in 2020. It's still not there yet, right? So how do we classify this into Chihuahua and muffins, right? So that's what there are two. I told you there are two approaches to machine learning. One is supervised learning, and other one is unsupervised learning. And the types of machine learning approaches, they come under these two categories: classification and regression. Again, there are two types. So in the earlier picture I showed you, we're trying to classify these images as whether it's a dog or a muffin, right? That's a classification problem. And if there's something that goes, increases over time or increases over length, you're talking about regression problems. So again, ML approaches can be divided into two parts. One is regression approaches, and then classification approaches. And then between these two major categories, you can have multiple subcategories. So for example, attribute ranking, clustering, and anomaly detection, and feature selection and association. So these are all different subcategories under these major types of ML approaches, okay? And what are the algorithms currently people are using? You must have heard about linear models, nonlinear models, dimensionality reductions, clustering, classification, nearest neighbors, SVMs, random forest and decision trees, artificial neural networks, deep learning. So all of them are different approaches to pretty much do the same thing. For example, SVM, random forest, they all do the same thing, classification. They classify into two categories, or more categories, the number of categories is defined. So is there a master algorithm? Is there something that is universally applicable? Is there something that is more powerful than everything else? And no, because the machine learning approaches can be divided into three parts. You're representing something in one framework and you're evaluating it, and then you're optimizing it. So it's just that. And each one of these approaches, whether it's symbolist approach or Bayesian approach or connectionist approach using neural networks, they have all their positives and negatives. So the way optimization in one methodology could be good and easy and may not be informative, right? So all these things are very important to understand, that there is no master algorithm, and you have to use and train and test your own algorithms based on your data and the questions that you want answered, right? So why machine learning in biology? I pretty much explained, the background, why why it is important and why it is useful. I don't think you need to be convinced anymore. But I just want to give you some real life problems, right? So if you just take two people's genome and just look at the differences between, what are the SNPs, the differences between two people, you'll notice that there are at least 3 to 5 million variants, right? And in the disease context, we're talking about, in the non-coding regions, it's 4 to 5 million variants, and 22,000 coding regions itself. Now, a pharmaceutical company or a clinician or hospital will have one drug to target one of these variants, and we don't know how to shortlist them, how to prioritize them. So that's where in our lab we use machine learning approaches, because we just don't know how to zoom in on that one thing that we have a drug against. So you can see why ML approaches are being useful here. So that one particular variant that is important is what we are all searching for in our lab. Now, if you take even one protein, right? For example, this is p53, which is one of the most compromised proteins in all possible cancers. You will notice that the red and green dots are indicating that this is these are all variants on p53 that has been identified in all these cancers. So you'll notice that it's not just one mutation, right? There are hundreds of mutations on p53 itself, and depending on the cancers, mutation patterns are different. Again, if you have one drug, how are you going to target the drug? How do you know where to target it, right? So that is where we use, and that's just one example. And how do we classify which of these mutations is pathogenic or benign? And there are no classification strategies. People use different classification strategies. For example, there are 8 classification strategies that people use to say a mutation or a variant is benign or pathogenic or not pathogenic. So that's what we are struggling with. It's not about the data, it's about the interpretation of data, and that's where we are using machine learning approaches. So that's one thing I told you. The one problem is how to prioritize variants among these millions of variants. The other one is, I briefly alluded to earlier, the majority of the variants are in the non-coding regions. We are just looking at 1.5% of the genome, and that has only a small fraction of the variants or mutations, right? So how are we going to interpret those variants that are outside of the coding region? So that's the two problems that we primarily work on, and we use ML approaches for that. And to give you a perspective here, even for these different cancers, 5 cancers that I represented, the blue bar indicates the number of mutations in the non-coding regions. The pink indicates the number of mutations in the coding regions. So you see how a very small fraction of mutations actually land on coding regions, right? So that is why we particularly use ML approaches in the lab to prioritize variants and mutations. So where do we start? So I told you data is being collected at multiple levels, whether chromosome level, at patient level, at electronic health records level, at epigenetics level, at all possible molecular details. So what is happening to all those data sets? It's all being converted into what is called a matrix, right? That's why matrix algebra that's where it's coming into ML approaches. So everything gets converted into matrices, and with that we start using them in our ML models, right? So before I kind of explain what are the different approaches and the details of those approaches, there's something that is fundamental and many people miss it, and that's what I put in a substantial number of slides just for that. And this is how do you clean up your data? How do you process your data before even thinking about implementing any ML approaches, right? And for that, you have to do this thing called constructing your data sets and transforming your data sets, okay? So that is the most fundamental thing, because everything else, every other interpretation, whatever model that you're using, fancy model that you're using, ML-based model you're using, depends on your data, the data that you get, okay? So right now, behind the hood, what's happening is you take a set of data and you call it a training data set, and you put in, you try to observe patterns from those training data set, And then after you observe and generate an hypothesis based on the training data set, you test the hypothesis on a test data set. So you partition your data set into two types, training and test. And sometimes people do partition into three buckets, train, validate, and test. So this is basically what's happening. They take a part of the data, train their model, and then test it on another part of the data set. So now what we're looking for when we're training and testing, right? When we are training and testing, we're seeing how good the data set explains that a particular hypothesis. For example, if you're fitting a line, is that error bigger? Can you reduce that error to a zero? And how do you do that? So this is test error, and an objective function of the test error is to minimize that error to zero. Okay, and when you do that, there are these two problems. One is bias and variance. And you can think of bias and variance as two sides of a balance. If you try to control for one, the other one increases, and if you try to control for the other one increases. So there is no there has to be a sweet spot, and you have to identify that sweet spot. You can never get both of them ideally to be zeros or in an ideal regime, right? You will either get overfitted model or underfitted model, and you have to always balance between these two extremes. Okay, so let's get back to the data, right? So the quality of your data is the most important thing that you should be worried about, not how clever your loss function is, how deep your network is, and what optimization algorithms you're using. It's immaterial. At the end of the day, everything is dependent on the data that you feed in, right? So those are the things that I'm kind of emphasizing here. So I'm just going to quickly go through all these slides because in the interest of time, but I can make these slides available to you guys to go and check out the details of what I've written here. So you got to clean up and remove duplicate values, remove bad features, and look for simple things like standard deviations and see whether there are any outliers. And you have to understand what kind of data set it is. It is a transnational log or attributes or aggregate statistics. And you also have to know whether your data is labeled or unlabeled, or sometimes you can actually derive labels. For example, you can add label based on two features. You can call it x plus 1 is column 1 plus column 2 is column 3. So something like that. So you can actually understand and see how much you can first pre-process your data sets before you put it into any of your models. Okay, so this is a fundamental aspect of ML approach. It's called feature engineering. So you're converting raw data into machine learning format, machine interpretable format. For example, you're converting some numbers into zeros and ones. And here you're converting numeric values into easily interpretable values here. For example, the number of rooms, you're converting it into a vector here, 6. And you can also convert categorical values, like for example, the street name into zeros and ones. This is all done by what is called one-hot encoding, right? And it's important to know how spread out your data sets are, values are. For example, you need to figure out whether you can actually constrain and normalize it within a particular range and normalize all attributes on all features within a particular range. Because if you don't do that, for example, if something varies from 1 to 1,000, and another of your variable varies from 0 to 1, when you're trying to optimize it, the computers, the algorithms, optimization algorithms will not know whether to go after the one that spreads out from 0 to 1,000 or the ones that goes from 0 to 1, right? So both of them have their own problems. So for that, you try to normalize them, put them all in one scale. So these are all the same points that I just mentioned earlier. I try to remove outliers so that you don't get your data skewed up in one particular distribution. So removing outliers using clipping methodologies, and you can bin them in equal bins or quantile bins, and make sure that you scale. I said there are different scaling methodologies. You can use z-score scaling, log scaling, linear scaling. And I also told you why it is important to scale. And even categorical values you can actually scale based on what is called sorting, vocabulary, and hashing. So you can actually put them into different numbers and classify them. So for example, occurrences of man, woman, car can be hashed. So this I mentioned earlier. So after you pre-process your data sets and you're happy with the way you did normalize it, remove the outliers, and did all the things that I mentioned, and you're going to partition it. You're going to partition it into training set or test set, and then you're going to train your model, like if it is dimensionality reduction methodology, or if it is a linear model, you're going to use that hypothesis and train using your training data set, right? And then get some errors, try to optimize those errors, and then test that model on a test set. So this is the second approach where people sometimes use what is called a validation set. So here, instead of, they train it on a particular small set of data, and whatever parameters that they call, these are called hyperparameters because the model has some parameters, and you tune those hyperparameters using a validation data set, and then confirm the result on a test data set, right? So there are three things. There are things that you have to keep in mind. There are too details. There are too much. I don't want to go into depth of these things. Imbalanced data sets and downsampling and upsampling. You need to think about how you can, what do these terms mean before even starting evaluating your models and assessing your models, right? So there are things called validation approaches where you again partition your data set into two, and you do what is called leave one cross validation, where you leave one out in your training data set, train, and then put that back and retrain and see what that error is. And this is the K4 cross validation, where you partition your training data set into 5 or 10 or whatever, and look at the errors and see whether those errors minimize as you randomly go through your training data sets, partition training data sets. And this is bootstrapping, where you take multiple small populations and check whether those errors can be minimized or not. So all these things are important to understand because this is still we're not there to even use ML approaches yet. We're just kind of talking about what to do with the data and how to pre-process them and how to use them, right? And things that that you have to keep in mind. Okay, after you have an ML approach, and how do you evaluate ML approaches, right? How do you know one model is better than another model, or one set of parameters is better than another set of parameters for the same type of model? So there are different methodologies to actually look at and evaluate your models. They call accuracies. You must have heard of, people talking about something is 97% accurate, something is 95% accurate, and for me, accuracy doesn't actually tell much. Precision recalls are the best way to look at how good a model performs. And even better, there's another score called F1 score that actually tells how good a particular, it's just one number, one metric, a particular methodology or particular ML approach is, model is, right? So the details of all these metrics of accuracies and ML approaches and models can be, you can find it after this talk and dig into this, what exactly they're talking about. There are other metrics too. For example, runtime, science of applications, right? For example, if a particular model takes 3 days to run instead of 5 minutes, there is this trade-off between time and how much accuracy you want. So you can also evaluate based on runtime and also the size of application. So if something that can run on a small iPhone app, compared to, if it needs a huge cloud computing interface. So that is also something that you have to keep in mind. Let me skip all these bias and variance because this is another set of metrics that you have to keep in mind while you're tuning your hyperparameters, while you're seeing whether your models are what you want from the models, because you're always going to either overfit your model or underfit your model. Okay, so the bias and variance numbers can tell us exactly whether you're where you are in that sweet spot. So having told you about how to pre-process your data and how to implement different methodologies to partition your data set and how to cross-validate them, partition even the training data set to proper cross-validations. I've also told you what are the metrics that you use to evaluate ML models. So I haven't told you what actually different types of ML models that people use and what we use. So these are different types of ML models. So one is linear models. These are basic regression, right? Logistic regression and linear regression. Cluster analysis is K-means clusters. And classification, I told you, it's just classifies into two bins: neural networks, decision trees, random forests, support vector machines, naive Bayes. They're all classifiers. And what do they actually do? So let's get into some nuts and bolts of what they actually do. So the dimensionality reduction methodology that I told you. So I told you that there are two approaches, right? The supervised methodology, where the data sets are not labeled. You're asking the computer to find patterns. So these come under this dimensionality reduction approaches, where you just don't know whether there are any patterns. For example, you throw in a set of 22,000 genes in 5 different conditions, and you have no idea. They're not labeled, and all you're asking the computer to do is just find patterns. I don't know what is happening here. So you use dimensionality reduction methodologies for this, and there are two quite highly used dimensionality reduction methodologies. One is principal component analysis, other one is TSNA. Principal component analysis is a linear dimensionality reduction methodology, and TSNA is a nonlinear methodology which can take in high dimensional data sets, like single cell data sets, thousands of transcripts, expression values of thousands of transcripts, and thousands of cells, right? And you might feel it's all quite daunting, right? You might, it could be intimidating. But actually, when you implement all these things, it's just one line of code in the current scenario of how these MLM approaches have been developed using in Python or in R. So the pseudocodes for principal component analysis is written here, and for TSNA is written here. So although it looks quite daunting, the mathematics behind it looks daunting. At the end of the day, when you're implementing all these things, it is just 4 or 5 lines of codes in R or in Python, because most of it is abstracted out from us. All these optimization algorithms, all these tuning of parameters, we don't have to really worry about it. And none of you should feel intimidated. You can just go in and dig in and start working. So this is clustering, k-means clustering. So what happens here? We saw that we're trying to ask the computers to find out whether there is any clusters in your data sets. You can see those clusters, you can not see those clusters, you can ask it to build based on hierarchical ordering. And classification, both random forest and decision trees, what they do is primarily take a data set and split them and keep splitting them. And depending on each splits, we can get good answers. For example, here, I'm just splitting a class of kids, and some are interested in playing cricket and some are not interested in playing cricket. And what split actually tells us, what decision actually tells us who is more likely to play cricket. So this is what is being shown here, and the cost functions is based on these 4 metrics, and we can go in. Sorry, I don't have time to go into the details of all these 4 cost functions. But what you're trying to do is you're trying to see whether you can minimize this cost function and see whether you can predict who is more likely to play cricket. So that's what decision trees and random forests are able to do. So this is a linear model, a simple linear model. Most of you must have done this in primary school or secondary school or high school. All it's trying to do is fit a line, right? And this is your hypothesis. This is your machine learning model, right? Approach. You're saying I'm using a linear model, that is theta 1 of x plus theta is my hypothesis, that is the y or h theta, and fit a line. And the cost function for this can be arrived analytically, but this is the cost function. So it's just minimizing the errors. So ideally you want a 0, right? Because you don't want any errors. So the computer will go through multiple iterations of theta 1 and theta 2 to find out the best fit line. Okay, so that is pretty much what's happening. So the cost function for here is very intuitive and easy to understand. So y equal mx plus c. And likewise, the cost function for all these curves are also easy to implement because it's just one line of code called LM function in R. And suppose if you are not interested in one fit, if you want to try out multiple, curves and see what's the best fit curve, and you want to give a probability estimate, you use Bayesian polynomial regression. Okay, so this is the Bayesian approach. All these terms looks quite intimidating, but at the end of the day, it's very easy to understand what they're actually doing. What line fits in here? You're not interested in one line, you're interested in many lines, and you don't want in one particular line, you want just confidence for many lines. So that's what it's doing. Logistic regression, again, it's one line of code. You want to choose whether it's, these are the different equations. You choose the equation, train it on a particular logistic regression model, you feed in your data, and just say find out whether this is the right one. Support vector machines, classifiers, they try to draw hyperplanes between two data points, sets of data points, and they will find hyperplane, and that's what they're trying to do. And again, it's like one line of code. This is neural network, where you have a simple architecture of one single layer of neurons, and each of these neurons are taking inputs. This is all your data sets on your left-hand side, the x1, x2, x3. And the data sets are fed into different neurons, and these different neurons take in and put weights for these data sets depending on what they want. And this simple one-layer neural network has this kind of cost function. Again, this is all arrived at analytically and solved, but you don't have to sit down and work it through, because when you just implement this model, there's underneath the neural network one line of code, all these functions are created for you, and somebody's trying to optimize it. And that has been all abstracted out, right? And you can create simple architectures, neural network architectures in R and Python, simply just by saying, okay, I want this number, this is the activation function. There are different kinds of activation functions, and I want to give, it'll arrive at itself in different ways. You just create this architecture like this, and 4 lines of code, you're just done, okay? And this is convolution neural network. You must have heard of convolution neural network. It's primarily used for imaging, where people abstract out parts of an image and save that as some kind of interpreted data point, and they walk through along the entire data set and keep noting that as some interpreted value, but we won't know exactly what it is. For example, if the digit 9 is scanned and the computer is asked to recognize the digit 9, it will just go through different parts of 9 and store it in some format, and we will not know what it is actually storing, whether the curvature of the 9 or the different aspects of how bright or dark it is. So we don't know, but it'll save it, and then it will try to interpret 9 whenever it sees it again. So these are, given all these different approaches, right? So I told you there is no one single answer, and there is definitely not going to be one straightforward way to tell that whatever you got is correct or wrong. So for that, you implement multiple methodologies and see which methodology is giving you the right answer. Again, machine learning approaches are primarily used for predicting, right? You kind of say, can I predict something in the future? But it is very dark and dense when it comes to interpretability. You can't interpret these models very easily, and that is the problem, and that's the main problem. You can get good classification, but you don't know why you got that classification. So that is where the field is. And there are some new developments in the field, causal inferences and other things that then enables us to do, but we're not there yet. So what would you do if you just have a straight line? You can see immediately that you can actually fit that straight line using all the possible approaches that I actually mentioned and talked about in the previous slides. Linear models, quadratic models, confidence intervals, logistic regression, connecting. It's just the same set of data points. So you have to be very wary of what models and approaches you're using, because anything that you use, you can actually get an answer. Okay, that's what I wanted to emphasize, and this all depends on the data. So that's why I started off my talk saying that your data is the most important thing. You have to pre-process it and make sure that the data is clean and doesn't have any artifacts, right? Are there any real examples? So I think I've covered different aspects of ML models, but now let's see where it's being used. I'm going to just quickly go through it. And I told you in our own lab, we use it for 2 different methodologies, 2 different problems. One is to solve how to prioritize mutations that map to protein coding regions. So I showed you an example of P53 where there are so many mutations and we don't know which one is important. So we use ML approaches to say, okay, now given this mutation, is this mutation important or that mutation is important, given we know so much about this, like biochemical features or its protein-protein interaction profile or biophysical properties. So that's what we do. And we also look into non-coding regions and certain non-coding regions that actually make protein products that can fold and form proteins. And again, you see these are many mutations mapping to those protein products in the non-coding regions, right? We develop classifiers and try and develop different methodologies to see how to interpret those regions. Other people have done for transcription factor binding sites, they've taken a set of known transcription factor binding sites and trained those models and see whether they can predict all possible transcription factor binding sites in the entire genome, or they can predict a new set of transcription factors or binding regions. And some people have looked at pseudogenes, some people have looked at promoter regions and checked out how to prioritize mutations in those regions. Right? And some have used it to annotate genomes. For example, if you know certain regions of genomes code for proteins or used for regulatory regions or bind histones, can you use those models, train them and understand them, and then scan the whole genome and predict more sites? DNA accessibility sites have been used, like, for example, all these new kinds of seeks that we are hearing about, high-seq, chip-seq, all those sites, the experiments can only yield so much, but based on the experiments, can we predict more such binding regions?
Okay. Dr. Prabakaran, sorry to interrupt you. We are running out of time, so you can.
Okay, so I'm going to just quickly come back. So I'll make these slides available. Again, used variant collars in ML approaches, predicting somatic mutations and how some mutations kind of correlate with expression values of genes and protein. How do you understand mutations in proteins, mutations in enhancers and promoter regions, and how do you fold proteins? Okay, this is Google's the AlphaFold, and other aspects as well, and drug discovery pipelines. And more than that, patient treatment as well. So a lot of resources here. It's all useful for you. You can take it from there. But coming back to COVID, right? So has AI actually helped? ML approaches have helped in COVID at all? Yes. In fact, in December itself, 2 companies actually predicted that this is going to be a major pandemic. And Meta Biota and Blue Dot predicted this, and using data sciences and NLP, natural language processing, meaning they've looked at Twitter profiles, Facebook profiles, and people moving, and based on that, they predicted the exact number of deaths in different months, and they were spot on. And the data for COVID is also available from these places. I would like you to take a look at this particular next strain one. It has real-time data on mutation profiles. And Uri Alon's group has come up with this fantastic adaptive cyclic extract strategies based on these data sets. And yesterday there was a paper in Nature Medicine which showed that 2 million people using smartphone apps and describing their different symptoms, and that can actually tell us whether somebody accurately, somebody is whether infected or not, even without a diagnostic test or kit. Okay, so and we have come up with we just published a paper saying that there are a lot of barriers and how to overcome those barriers to be able to use these different data sets and to be able to help patients and give prediction based before something can happen. Okay, so with that, all these ML approaches I talked about, we actually do it as a 3-day course, and these data sets and details are all available in this GitHub link. You can go there and run and train yourself, and it's just easy to use. Okay, and I just want to thank everybody for the opportunity, and I hope there was some take-home message for everybody. Okay, I'll take questions now. And Shailesh, sorry for running over time.
So thank you, Dr. Prabakaran, and thank you for the very informative and interesting talk. It really enlightens many of the participants how data science can be used in biology. And really it's a very interesting, because you started from the history, dealt with the big data problems, machine learning, and how it can be used in genomics. So let's have some questions. Before questions, I just want to everyone that your mail.
Sorry,
just
can you please repeat that?
There will be one feedback form for the participants. They have to fill it after the talk. So this is for the participants only. So let's have few important questions. There are a number of questions which we have received, but two questions which are very interesting. Like one question is there, what is the difference between forecast and big data?
Big data is
Is there any difference between?
it's just buzzword, right? It's just data sets, but data itself doesn't tell you anything. As I told you, you have to find patterns, and those patterns will tell us and will give us the ability to forecast, right? And data is data, and ability to forecast is what we have to do with the data. So that's the difference.
Okay, thank you, sir. There is one more question which is related to COVID-19. So that is there any way where we can say that by extending the lockdown, we can kill the pattern of spreading the infection? I mean, in an Indian perspective. So what's your idea by looking at the Indian data?
So
Yeah.
that's what, so there are different models out there. As I told you, even for the Spanish flu, different cities implemented different approaches and they saw different responses. For example, Philadelphia was very late to implement, this was for the Spanish flu. They saw a huge spike in the death rates, but then they understood what happened and then they clamped down. And likewise, even now, different countries are implementing different methodologies and nobody's successful. At least some countries are successful in the curve that I just showed you. But what is important is you should also check out Uri Alon's it's a fantastic model where they've actually told in a cyclic way, you open up open up the lockdown and let people in to work and first initially for 5 days or for 3 days and then 4 days and then 5 days and then different groups of people, and they've shown that this is more effective than clamping down completely and opening up one fine day, right? So that is what they have shown, and I think that's what is going to happen. It's going to be staggered, and that staggered has to be based on some model, and that's the model that people are trying to come up with for that particular city and for that particular state and country. But it's important to understand one thing. Because people move between cities and countries, it's very difficult to implement one approach and say, okay, this is good and I'm going to stay with it, right? You've got to keep calibrating all these things constantly. Yes, cyclic methodology is the best way to forward, not long lockdown, because unless you have vaccine, you have to wait out until you get a vaccine, but that's not going to be helpful either. So you need to make sure that there's some amount of cyclically phased out lockdown.
Yeah. Thank you, sir. There is one more question which we can take. That is, how you are dealing with the parameter tuning in this ML approach. Is there any way how
that's an interesting question.
Can suggest any?
Thank you. That's an interesting question because that's what a lot of us spend time on, because we don't know what parameter is important, like L1 regularization, L2 regularization, different methodologies. To sometimes you can do what is called permutation imputation, like remove one feature completely and see how much it disturbs the error of the error function. And how much it is important. So not all parameters will be important. Some parameters will have some weights. Some parameters will be less important. So it's very difficult to interpret that way. So we all struggle with that problem. And the only way to do it is some kind of combinatorial methodology and remove some systematically and change the weights and see. There are frameworks for doing all these things.
Yeah. Thank you. One last question that is very basic one. Which language is best to learn this machine learning approach in biology? Is it R, Python, or what? What's your suggestion regarding this?
That's an easy question. I would say start with R, but you have to go to Python. That's R is very easy to implement, and a lot of packages are available easily. Python is also easy. I would use Python, but to begin with, to get your hands wet, you should start with R.
Okay. Thank you. I think we have 1 more minute to ask one last question. That is there any software for find the mutation between coding and non-coding region, or is there any way to do it? Means this was the question from one participant.
There's no software. I mean, because so far people have been doing only whole exome sequencing, where you just get a list of mutation for known genes. Now people are doing whole genome sequencing quite regularly, so they get mutation profile for the entire genome. So there is no software. We just write our own programs and we remove the regions that we code for proteins. That's how it's done.
Okay. Thank you, sir. Thank you very much. And really very interesting talk and very informative also.
Thank you.
Thank you all participants for joining us in this webinar. I would like to inform you that in the series of the webinar on data science, we have Dr. Krishnadev. He will be talking in the next in the series on 21st May. It will be fine if you can find information regarding this on our social media platform like Insta, Twitter, and Facebook. So I thank everyone, especially Dr. Prabakaran and team, technical team, whoever is working for this webinar. Thank you. Thank you, sir.
Thank you. Cheers. Bye.
Thanks.